Bom dia a todos. Hoje a palestra da disciplina de Seminários de Zoologia do curso de pós-graduação em Zoologia do Museu Nacional e do curso de graduação em Ciências Biológicas, ambos da UFRJ, será sobre filogenômica e a árvore da vida dos anelídeos, ministrada por um dos expoentes do tema, professor Christoph Bleidorn, da Universidade de Göttingen, na Alemanha. It's a great honor to have Dr. Christoph Bleidorn here with us to talk about phylogenomics and the annelid tree of life. Thank you very much for accepting the invitation, Dr. Bleidorn. He has received his PhD from the Free University of Berlin in Germany in 2004. After this, he has had positions as researcher and professor at the University of Potsdam and the University of Leipzig, both in Germany, and at the Museo Nacional de Ciencias Naturales in Madrid, Spain. Since 2017, Dr. Bleidorn is a professor and head of the Department of Animal Evolution and Biodiversity at the Georg-August-University of Göttingen in Germany, and he has an outstanding career with influential studies on evolution of Annelida and Spiralia, arthropods and their endosymbionts, phylogenetics and phylogenomic methodological and theoretical issues. These studies have been published in high-impact journals such as Nature, Molecular Biology and Evolution, and Genome Biology and Evolution. And he also has a very enlightening book on phylogenomics, an introduction, published by Springer. Dr. Bleidorn's main research interests are evolution of arthropod endosymbionts relationships, annelid evolution, and evolution of the venom systems in glycerids annelids. So thank you very much, Dr. Bleidorn, and without further ado, Dr. Bleidorn.
Okay, thank you very much, Joanna, for the nice introduction and also for the invitation. I'm very happy to give a seminar in Brazil. So I was also like putting a nice shirt for the Brazil spring on here because I guess it's nicely warm. It's a bit rainy today, even though it's also warm in Göttingen. And I want to talk today about the phylogeny of annelids and also what happened in phylogenomics. So, and I will basically give you an overview about what we learned about annelid phylogeny in the last 20-25 years and where we are now and what could be the direction in the future and what does it mean. But also I want to show you a bit how the methods developed and how this helped us to unravel the phylogenetic relationships of annelids. So annelida, as you know, they were like traditionally separate or like distinguished in two big taxa: the polychaetes, the polychaeta, which we know nowadays are paraphyletic, or what you could do, you could just synonymize annelida with polychaeta, then they would be fine again. Which I usually call them the marine annelids, even though they're also cladodates, I know. But they are like very interesting forms. Some of these forms were described as sedentary forms, like you see here, or here in this Catopterus, and some of them were called errant forms, which were much more vagal, often predators, or like often could move very well, like the nereid you see here, or the psyllid you see here. And then on the other hand, there were the cladodates. Yeah, the cladodates is a group of annelids which include something which we can briefly summarize as leeches and earthworms. Yeah, and either one or the other way of living is quite well characterizing the biology of most of the taxa in the cladodates. Yeah, and then, of course, there were several groups around the annelids, which were most of them were first put in their own phyla, yeah, like the Archaea, the spoonworms, the serpunculids, or also the mysostomids, which were like moving around the tree of life since they were described. And there was always a question of these animals: are they annelids or not? Yeah, mysostomids, for example, were one of my favorite taxa of annelids. They are like symbionts or parasites of usually echinoderms, usually crinoids, and I guess Archaea and serpuncula are quite well known. These are annelid-like invertebrates which do not show very obvious signs of segmentation, and but often have like very similar forms of living. And then when I started my work on annelids, I was a diploma student in the group of Thomas Bartholomäus in Bielefeld at that time. And there was a huge question which was posed nicely in a paper by Wilfried Westheide. And there was a question about what is the direction of annelid evolution? Yeah, because we have all these different forms, and we have like the clitellage, which are usually quite simple organized. So they don't have what other marine annelids have, like obvious parapodia. They only have few, not very complex chaetae. They lack completely any head appendages, for example. And then we have the what we call polychaetes, which is like a mix of simply organized forms, but also very complex forms in a way of morphology of the morphology, like having obvious parapodia. They can have head appendages, maybe they're building tubes. They can have this what we call elytra here in the polynoids. And there was always a question: how did the last common ancestor of annelids look like? Was it something like simple organized form like the clitellades, or was it already a complex organized form like we see in many marine annelids? And then, of course, it would mean, in the one hand, often what people often assumed in earlier times, like there's a progression and evolution and things getting over time more and more complex. Or maybe we started with a complex ancestor in this case, and over time there was like many reduction in many different lineages, so that the simple forms evolved from complex ancestors many times. Yeah. So here complexity is always measured in like morphological diversity we can describe. Yeah. And that was a big question at the time. And there was one very influential paper in 1997. So some years ago, there was no annelid talk without this phylogenetic trees by Greg Rouse and Christian Faufeld. And what they did, and that was very good at that time, is before they started with this kind of analysis, most of these phylogenies were more like anecdotal. And they said, okay, we need like to use a precise cladistic way of analysis. So basically what they did, they used all the data from their own observation and literature, and they made a big data matrix, and then they used like phylogenetic methods like parsimony to reconstruct the tree, and this is the phylogeny they came up with. Yeah. And they really found, we have the outgroups here, which also included sipunculates and Archaea, and the clitellades are the sister toxin of the polychaetes. And if you have here the sipunculates and the Archaea, you would basically assume that the scenario that we had a simple ancestor and the complex forms evolved later would be quite fitting. Nevertheless, this publication was, or this tree was discussed from the very beginning. It was published. And at that time, there were only morphological trees, no molecular trees, but also the morphologists, they were hardly arguing basically about every single entry of this matrix, which of course can have influence of the tree. And so it was always called in doubt if this is really representing phylogenetic relationships of annelids, or if this should be reanalyzed and reconsidered. Okay. That's when I started working on annelids. I came to the group of Thomas Bartholomäus, and they needed somebody working with molecules. Even though I had no idea how to do this, I said, yes, let's do this. And I started to work in my diploma thesis on one molecule to resolve annelid relationships. And that's what you did in the end of the 90s, in the beginning of 2000. At that time, when we said sequencing, there was only one way of sequencing, that was Sanger sequencing. At that time, it was basically a synonym. People were saying sequencing, were talking about Sanger sequencing, yeah. And there were sequencers like what you can see here. This is an ABI. There was like not that many different brands on the market, and they, with this technique, it was very feasible to produce like, I don't know, like overnight, maybe 100 sequences of 1,000 base pair lengths. And at that time, it was a bit more expensive like now, but nowadays I don't know, you pay per sequence like €2 or €2.50, something like this. So you could, with a few genes and PCR products, you could do quite a lot of sequencing. And what I did in my diploma thesis was, not in the diploma thesis, in my PhD thesis, in one of my first publications, is that a lot of annelids which were collected in this group I produced the first 18S sequences. It was at that time the go-to gene for making deep phylogenies. And then we were basically saying, okay, when we analyze this 18S gene, it's a ribosomal RNA, there were like many, many good primers available to sequence it. What does it say about annelid evolution? Unluckily, it wasn't saying much, because when we were analyzing just one gene, we found that the monophyly of many families was well supported. So there's like a taxon category called family, which all the polychaetes and annelids are sorted into, and many which was described on this level seemed to turn out to be monophyletic groups with some few examples, and that was then interesting what we could found. Sometimes we found, oh, this genus belongs in this group or not in that group or something like this. Nevertheless, the relationship between these families remained completely unclear, because the support and the resolution was not very well. So even with this 18S molecular tree, we were basically not much, didn't know much more about annelid relationships. And but we also didn't find support for any of the higher taxa proposed by Ross and Fauchert. But of course, this could be connected because maybe the information was not enough in this gene. And which was also not nice, the annelids were not even recovered as monophyletic. And that is not because groups like Cypunculates and Archaea, which we expect to move into, grouped in there, but also some mollusks and whatever. So it was like showing us with this one gene, we are not getting there. We cannot just take this one gene and resolve all the problems we have with annelid relationships, even though it started to help to get us into this direction. At that time, it was like in the beginning of the 2000s, we were far away from this phylogenomic age. So basically what people were looking for, it was the golden gene, yeah? Everybody was looking at different genes and trying and looking, if we analyze this gene, maybe we got the super well supported phylogeny of everything, which of course wasn't working. So people tried with 18S, 28S, elongation factor 1 alpha with the protein coding gene. And of course, you can even combine all these genes, and you have 3 genes. And what you found in this analysis was often the same picture, that the monophyly of what we described as family traditionally was often well supported, but the relationships of these families remained unclear. But there was also no support emerging for the groups proposed by Ross and Fauchert. At least we found now, when we were combining 3 genes like monophyletic annelids, at least when we include Cypunculates and Archaea. Okay, but at that time, it was like in the mid-2000s or so, there was a new era beginning. And at that time, there was some influential paper coming out, and one of this was by Antonis Rokas, was published in Nature, genome scale approaches to resolving incongruence in molecular phylogenies. So that was exactly the question. So we were looking at one gene, got one tree, we were looking at another gene, we got another tree, and so on. And what they did at that time was, at that time, there were not many, for example, in annelids, at that time, everybody knew by heart which genomes were sequenced. It was like 4 or 5 genome sequences. But there was like in the yeast, yeah, Saccharomyces, there were several genome sequences because they were quite small and they're like model organisms. They're also like very important economically. So there was a lot of data of this genomes, and they combined it and looked what happened if we look at single genes picked from this genomes, from all this yeast taxa, and analyze it. And then they found that like different genes were supporting different topologies. And then they combined everything in one big analysis. And what they found is that they found a perfectly resolved and also perfectly supported tree with high bootstrap support, Bayesian support, when they combine all the 106 genes. And then of course they were making like some estimations. They found it doesn't matter which 20 genes you combine from these genomes, at some point you always arrive at the same topology with significant support. And that was the message with Henry G. was like writing an editorial in Nature, or like a comment, this is the end of incongruence. Finally, we have it. You're not getting anywhere with your few genes. You have to combine them for phylogenomic analysis to make finally we get the true tree. Yeah, something like this. So we will never know if it's a true tree, but something like this. On the one hand, this was good news because, okay, we are getting there. On the other hand, in 2003 or 4, when this was published, it was also a bit of bad news because I'm not getting so I'm not getting 100 genes for all the annelids. I'm not even getting 20 genes for all the annelids. It was like basically impossible to do this in a reasonable number of time for like a huge number of taxa. That was a problem. So it was good news, but also, yeah, but not for us. So the question is, why can this happen? Why are like different genes supporting incongruent trees? So one of the problems what can happen is you have the violation of the orthology assumption. So what can happen is that, so when you look at the homology of genes, yeah, there are two different cases, three different, but I start with two. There are different cases which can happen. Genes can either be orthologous or paralogous. So if they're homologous at all, which are just decided on sequence similarity. And in the way a gene is split in its genealogy by speciation events, we talk about orthologous genes. If it's split due to gene duplication events in a single lineage, and which is then given to that ancestor or descendants, we talk about paralogs, yeah? And it's very easy. So what people hope is if we reconstruct all the orthologs which are available, we reconstruct the speciation history, and then we should get end up with a species tree. But in reality, it's often not that easy to distinguish orthologs from paralogs. I don't get into the details now, but you can ask me. And this is like a reason why you maybe end up with problems that that we get wrong phylogenies, because maybe we are analyzing a mix between orthologs and paralogs. Reasons for this can be gene duplication, horizontal gene transfer, lineage sorting, whatever. Yeah. So just to spoil this a bit, it's not that easy. It's not that easy that when you only work with orthologs that you always get species tree. That is another problem. But at least if you mix orthologs and paralogs, you're not getting there at all. Then, of course, this question of stochastic errors. If you only have one or a few genes, you have like sample size problems, yeah. And this stochastic errors due to gene length is like a problem. There was like a later paper by Antonius Rokas, and he was like trying to find what is the most significant correlative parameter which predicts the phylogenetic usefulness of genes, yeah. And they were looking at different parameters, and in the end it came out it's the alignment length, yeah. The more characters you have, the more likely is that these genes are informative. It's very easy. And so this is a problem. So if you have only one gene, obviously you have like very few nucleotides or amino acids you analyze. And that is like something, of course, a problem with single or few gene analysis. But so these two problems can be a bit overcome by phylogenomic studies. In the first case, it's because maybe if you make only a few mistakes, you don't see them in a combined matrix. Here, the stochastic error is very clear. The more data we have, the less likely is the stochastic problems. But of course, the problem is that you also have systematic errors. So these systematic errors are errors you have in your, which basically lead to artifacts in your phylogenetic analysis. Here's just a selection. You can have the heterogeneity of the nucleotide composition between different lineages, the compositional lineage signal. You have a variation of substitution rate between different lineages, the rate signal. So this is what you often see if you have these trees with short and long branches, and often long branches are maybe artificially grouped together, and even one long branch is difficult to place in any phylogeny. Or you have even this variation of the substitution rate within nucleotide positions over time. It's so-called heterotouchy. It's also a big problem. This often happens. What we have to assume when we use our models usually is like that the substitution rate we apply now was also valid like when we go deeper down in time in the tree. But this can maybe change over time. For example, if genes maybe are adopted in a new in a new way how they are expressed, and maybe they get more conserved, and then of course the substitution rates may change over time. So all these are problems. And they accumulate to something what we call non-phylogenetic signal. And the problem with this is the systematic errors, they're getting not better when you have a big matrix, because they're getting even worse. So something which is maybe not significantly supported in a analysis with few trees becomes like significantly supported in a big tree. Yeah. One caveat, of course, everybody should know, the bootstrap support just tells us, is our like data matrix big enough to make this conclusion? It doesn't tell us about anything about the truth about any nodes. Yeah, this is often what is misleading. And you're just getting more of this wrong signal, and you're getting maybe. So other people were claiming maybe the phylogenomics, in fact, could be the beginning of incongruence, and that is the reason why you have to deal later on with all these problems. So basically, you make a phylogeny, and then you make many, many different tests to see if this is a problem, is this is a problem, is this is a problem, is this a problem, and if always you come back with the same result, you say, maybe we are fine, this is now the best supported hypothesis we have for specific relationships, and let's see if they stand the time. Okay. Phylogenomic analysis of hundreds of genes. So we started with this in the mid-2000s. And like as recommended by everybody, there was a huge project in Germany called the Deep Metazone Phylogeny Project, where many, many groups started to get into the genomic age. The problem, of course, at that time was we started with this project like in 2005, and it was before this revolution of next generation sequencing really swept over the market. So it was not available, or it was not really available at that time to that everybody could use it. Yeah. So we were still sticking to Sanger sequencing. So what we did at the beginning was phylogenomics with Sanger sequencing. When you think now about it, it's completely crazy. So, but how do you do it? So instead of going there, making primers and like I have to amplify this gene, this gene, this gene, and so on, which will be like a lot of work and often impossible. What we did is like the reverse way. What people are doing is they were working with RNA, and they were like extracting RNA from the organisms wanted to sequence, and then they were only concentrating on the mRNA. It's a very little part of the complete RNA. Most of the RNA you find are ribosomal RNAs, and this messenger RNAs are basically transcripts from genes which are expressed. And then you took this messenger RNAs from this RNA, like rewrite this in cDNA, and clone this into a ligate this or into a clone, yeah. And then we grew them in bacteria, and then we could pick different clones. And the more clones you pick, the more different mRNAs you maybe find. It's a top-down approach. That means the higher this gene was expressed, the more mRNAs you will find, yeah. But that would mean that usually this kind of housekeeping genes, which are like highly expressed by all animals, there's like a certain set of genes you would expect to always find, like ribosomal proteins. You will always find some with this if you pick enough of these clones, and then you can up with big data matrices. Nobody can, so we were doing this for annelids, so we only analyzed like, I don't know, 16, 18 taxa, which we use with this method. And we make this cloning, and we only picked like 1,000 clones, but even that would be 18,000 clones, and who did cloning in the lab, nobody of you want to pick 18,000 clones in reality, even though it could be possible. But we were lucky that we were supported by the Max Planck Institute in Berlin at that time, and they had a machine, they had a robot. They were already involved in the Human Genome Project, and they had these machines where you analyze this and can pick a lot of clones, and then we could make all this express sequence. So what we did is we could combine 231 genes into a phylogenetic data matrix, or in a data matrix. This data matrix honestly looked like a Swiss cheese because there were a lot of like holes in there, like a lot of gaps, because of course we didn't find every gene for every taxon we analyzed. Nevertheless, we had a lot of data at that time, unprecedented for annelids to analyze. And what we really could come up with in this analysis was the first phylogenomic analysis of annelids, is that we find a new backbone of the annelid tree of life. And what we did is we find two big monophyletic groups, which we called Errantia, which are here in greenish, and Sedentaria, which are here in bluish, with a sister taxa. And later on, Thorsten Struck calls them Pleisternlieder. And then we had some taxa here in the basal branching part of the tree, like Ketoptrids and Serpunculates. Yeah. This was very nice. At the beginning, people were very unhappy with that we used these names Errantia and Sedentaria, because these were like iconic taxa as examples for paraphyletic groups or non-monophyletic groups. But they were quite well fitting the description here. It was not completely one-to-one relationships, how they were initially defined by Fauvel. But in the end, I think it was quite good that we use these names, because everybody's using it, because people are used to these names. Often when you come up with new names, people cannot remember them and don't want to use, and that was fine. So that's the name now. And usually people use them, and it's fine. Errantia, for example, includes like the Neredids, like Platineres, and the Sedentaria includes, for example, the Orbinids we heard about, like Naineres. Also, of course, also the Clitolates, which is new that we push them into the. sedentaria. So what we found, titulates are highly derived annelids, which also was for many morphologists not a big surprise. Many people were expecting this. And also we get a well-supported tree. At that time, it was like really nice to see that people were very happy to have this tree, because there was like many evolutionary developmental biologists like Detlef Arendt who wrote like a comment on this paper. Say it's super good. Finally, we also can place like Platinereus phylogenetically into a tree. Before we didn't have any idea how about the annelid relationships, and now we can much better see how our results can be interpreted in like even. But of course, there are bottlenecks of Sanger sequencing if you really want to go large scale and analyze phylogenomics, because what we did there was like hugely expensive. Nowadays, you could like sequence like I don't know how many human genomes for this price, and also takes time, it's laborious, and you cannot really scale it to many taxa. One is the library preparation, how you do it for the Sanger sequencing, because you include PCR and cloning steps, they are time consuming. Then the template preparation, what you have. And the sequencing. The sequencing on the Sanger is done using a gel, and the gel is like a little capilla, and then the sequences are going through there, basically sorted by size, and then you see one after another the bases coming out there. And then with a fluorescent marker, you recognize which nucleotide is coming out there. But the problem is with this method, you cannot really scale it up. You cannot build a Sanger sequencer, which is like sequencing millions of sequences. There was like a limit. Yeah. So we can maybe get 1,000 sequences overnight in one machine, or maybe in an afternoon, but you cannot really scale it like to get millions or billions sequences of what we see now. But we were lucky to really live now in this revolution of the next generation sequencing. And this started basically after the first human genome was published in the beginning of the 2000s. And then were people asking, okay, now we have the human genome, what can we do with it? And then people realized, maybe not much. So it was already very interesting. It was a very important step. But they saw for all these questions about, for example, a relationship between like a disease and its phenotype and a genotype, we need many human genomes. We cannot use only one. We need like hundreds of thousands to really get meaningful analysis for this medical approaches. Yeah. So then there are people sitting together with like people from the National Science Foundation. They were asking, what do you want to pay for a human genome? And somebody was making a joke and saying $1,000 would be fine, which was really a joke, because we were that human genome, I think it was like in the billions, how it was like paid after what was this project over 15 years. Yeah. So obviously that was a joke, but they say, okay, we have to find new techniques to sequence, because with Sanger we are not getting there. We need new ideas to do this. And they were putting on a lot of funding for new sequencing machines and techniques. And the first one which came on the market was the WASH 454, which we know as pyro sequencing. It was already dying now in 2016. So rest in peace, pyro sequencing, which was the original next generation sequencing, which was with this machine was able that people sequence the genome of James Watson, so one of the quick Watson, and they did it for like more or less 100,000, which was already much cheaper, but not getting there. And at that similar time, there was also Illumina coming on the market, 2007. The old name was Solexa, then Illumina. And they had a so-called reversible terminator sequencing technique, which was like really like a game changer. It was so-called sequencing by synthesis. You were like incorporating nucleotides in your sequence lengths. You are sequencing one after another, which are chemically blocked. And basically what you do, you incorporate a nucleotide. It has like a fluorescence. You detect the fluorophore. You detect the fluorophore. You remove the blocking, and then you make. Before you do this, of course, you make a picture, and then you remove the blocking, and then you go on with the next one after another, after another, after another. And with this technique, now you can do this on a flow cell. I have a picture which you see here. It's like very old pictures. And every data point, which are like light signals, at that time they used 4 colors. Now they only use 2 colors, is basically one data point of a sequence. And with this techniques, it was first possible to have like tens of millions, then hundreds of millions sequences, what we are doing now in one sequencing one. Yeah. So instead of like having 1,000 sequencing accumulating for 100,000 base pairs overnight, we now get in 2 or 3 days sequence amounts which we measure in terabyte. Yes. So it's like really a game changer, and it was only possible because of this Illumina technique. And now this Illumina family also spread. So we have like different machines, the MySeq for the lab is having like a few sequences, or this HiSeq extended at that time. Now we have NovaSeq, where you can basically, what people were saying, sequencing complete continents. Yeah? That's what people do. And this also came to a drop of price. I guess you have seen this graphic many, many ways. It's basically forbidden to give a phylogenomics talk where you don't show these graphics. It shows the development of the cost of per raw megabase price of DNA. Yeah, and this is like what you see here is Moore's law. Yeah, that is basically how the computer chips are improved over time. And then you see here, this was like Sanger, got a bit cheaper. And then here was basically 454 invented and came on the market. And then the prices dropped per megabase DNA considerably. Then here's another drop when Illumina really took over. And then here were another drop when these companies get under pressure because there were more competitors in the market and they get even cheaper. And when we go down to this price now, which is like really incredibly inexpensive but you have to at least invest something to get this data. So even though the prices are like you pay for like, I don't know, like a cent for 1 million base pair, you cannot just buy 1 million base pair. That's a problem. You cannot just buy for 1 cent. You need to invest like, I don't know, several hundred euros always. So this is the price per human genome, what happened. And what happened is, yeah, instead of like 100 millions, we pay nowadays around $1,000 for a human genome in 30x coverage, which is amazing. So we basically come to this prediction. Illumina announced within NovaSeq in a few times, you maybe can get to the $100 genome, but I think we are not getting there. So sequencing now is not the bottleneck anymore. That was just recently in a Zoom meeting where people told me the problem now is to get like space. That the most expensive thing is the space to save the sequence data or to analyze it. Yeah. Now people have to pay like, I don't know, 10,000 euro for analyze it somewhere in the cloud because you have to rent it from Amazon because there are no capacities anyway. Instead of putting all this money in the sequence data itself. So this is an update to now that we are more or less around the $1,000 genome at the moment. And what we used at that time, which was now 10 years ago around, or like 9 years ago, we started with a project. We used this Illumina technique for analyzing transcriptomes, which is like so-called RNASeq, which is basically using this EST kind express sequence text, but with Illumina without the cloning. And we had a nice project together with Thorsten Struck. He already gave a talk, I think, in this seminar. Günter Purschke, who all the other people well know. Then Ken Halanic, I think he's giving a talk or also gave a talk, who likes to work in Antarctica. And we came up with a project to make like basically a huge scale phylogenomic study to include much more annelid taxa to get maybe to see if our tree holds. And this was mostly done by Anne Weigert. She was a PhD student at that time in my lab. So this is always what you do. You come up with a pipeline of your phylogenomic pipeline. We got all this Illumina data for this annelids, which now was much cheaper. So sometimes there's time. When we think about how much money we spend for the Sanger analysis, we could have now analyzed, I don't know, like 3 times the number of taxa than we had in this analysis. Then we assemble them, these transcriptomes, and we look for the orthologous genes with a method called hamster. Then we made a huge alignment matrix, which I will show you. We moved like ambiguously aligned nucleotides. We concatenated all these alignments. We reduced the matrix. I'll show you why. We looked for the best models to analyze it, and we analyzed it with maximum likelihood to get our phylogenetic hypothesis. So this is what the matrix looks like. Even this method was still so some of the taxa were newly sequenced, then they usually look like this. Some of the taxa we took from GeneBank or from our old study, then they had much fewer entries. And with this matrix reduction, what you're doing, you can use an algorithm to say, remove all the rows or columns from my analysis which have like less than this number of entries or so. Yeah. And then we can also do this in a different size, and you can allow to have more or less gaps the gene presence in your matrix and experiment. So this is like a nice and easy way to see, does the gaps in my alignment have an impact on my phylogenetic result? So that's what we did. We had like unreduced data sets where we had like more than 2,000 genes included. And then we have like two different subsets called 1, 1.5, and 2. These are different specific Kmere parameters. And then included more or less genes. So the less genes were included, the more strict we were removing genes which were quite gappy for the taxa we were analyzing. Yeah? So what it means here we have like only 300 genes analyzed, but they were present in most of the taxa. Here we have 600 genes analyzed, but there maybe were also a lot of missing data in this data matrix. And we wanted to see, does it have an influence? And we found, no, it doesn't. So we always come up with the same tree. It doesn't matter if we analyze 300, 400, 600 genes, which were well covered. And we were basically mostly supporting what we found in the study from 2011. We had to still see this Archaea here in the sedentaria sister taxa. But what we saw, what was very nice, at the basal part of the tree, more taxa were getting there because we didn't include them before. So we already had the catopterids outside this variation and the sarpunculates we already knew, but now we included the first time oviniids and magallonids and amphinomids, and they were also branching outside the main annelid radiation, I would say. But all what we found here was super well supported in boosted analysis, which of course doesn't mean if we have a systematic error that this can be an artifact. But we were quite happy with our analysis, and given on this NGS data, on this analysis of the matrix occupancy, we were also quite confident that this is not artifacts. And what was nice is when we look in, so we finally have like a very good backbone here on the annelid tree of life. And it was super interesting to see Ovinia and Magellona as sister taxa together as a sister group of all other annelids. Many people were like doubting this at the beginning because they say, but why not the sarpunculates? Why not the sarpunculates? They basically look like proto-annelids, yeah. But on the other hand, there were several people like Reinhard Rieger who were all the time saying this, look at the ovinids, look at the ovinids. If you look in the papers from Reinhard Rieger, for him, ovinids is like basically like the master plesiomorphic state of everything in many morphological characters. Maybe this could be also basally branching, and they are. So later on, we call this group ovinids and magallonids paleoannelids, and we find them as a sister taxon, and then we find the sarpunculates here, a sister to the amphinomids, yeah, and we find, for example, the accurates here. So what does it mean? That means that taxa like which we see many other examples can lose segmentation secondarily. So they maybe these are very good examples of taxa which show like a secondary morphological simplification. In the kind of sarpunculates, it's a bit still discussed, but for example in the Cure there's very nice data which is already 20 years old from the lab of Westheide from Rene Hessling, who found that they find kind of segmentation in the nervous system in the development, which later gets lost, and which they interpret that these are the remnants of the original segmentation, which is not there anymore. But if you look all over annelids, there are several taxa which are not segmented, yeah. But most of them are not like such prominent macrofauna, but often they are like myofauna taxa, for example, duodilids or so. Nevertheless, the ground pattern was very difficult to view, so we wanted to see how the last common ancestor looked like. But when so the taxa mostly influencing the ground pattern reconstruction is all the taxa here at the base of the tree, and they look completely different. We have something like the amphinomids, which look like Archaea, but not grouped within them. Then we have the magallonids, which look like the typical sedentaria, but also not grouped in them with this prominent pulps. Then we have catopterids, which looks like somebody invented this animal, which are living in tubes. And it was super difficult to make any recommendation about the ground pattern. So of course we can reconstruct how it looks, but often we have, yeah, maybe it has this character or not, yeah. So what was very nice to see two follow-up studies, one which was like also mainly from groups which were involved before, here led by Thorsten Struck, but also another one which was like led by the team from Greg Rouse, and we see here also Brazilian and also here Spanish players in the field, and they basically, the studies come to the same conclusion. So there may be like within the sedentaria, so there are of course still discussions which taxon is sister taxon of what, but the main like topology of having the Errantia, sedentaria, sister taxa, and then some basally branching groups, this is like always comes out the same. This is very nice. So we are quite confident this is like a very good backbone on the annelid tree of life. We recently found when we reanalyzed this with some more taxa that there are like some more groups joining at the base of the tree. For example, the apistobranchids is like the apistobranchidae, often people never see them in their life. We have found them in the Arctic and Greenland, and the samudrilids, like a myofauna taxon, they are also branching here. But generally, we find again and again the same pattern, which is very nice. So we were very happy to have like a solid annelid backbone. Yeah. So at least this big backbone is quite solid. With in-group relationships, we need just more taxa to analyze. Everybody waits for a study Ken Halanic already presented 4 years ago in Cardiff to get on with this, but he is so interested in Ctenophora that we never see anything getting published there. And the next question is, of course, how old are annelids? Because when we were publishing this paper, the studies, people often say, this makes no sense because the Sarpunculids, we already know them from the Cambrium, and all these other groups, they look like perfect, like the modern annelids, what you have there at the base. It's very unlikely, and we would assume something maybe look more like Errantia or so, because they are like fossils like Canadia or so from the Cambrium, which look like this. So if you look in the tree, Sarpunculids are at least 520 million years old. They're super well good fossils. That means that everything was branching off before here must have split here from this part of the tree more than 520 million years ago. But then there's the question, why don't we see this in the fossil records? Where are the ancestors of the annelids? Where are they? And here they are. This year, sometimes very nice to see paper coming out. People were describing fossils from the Cambrium, from the Shenyang fauna, I think. And they look like Magellonids. They look like tube-bearing Magellonids with super pipes. So maybe it's not that absurd what we find here, that this group split off from the rest before 520 million years ago. Yeah. So this is also one of the problems. Of course, you should never forget, if you're doing deep annelid phylogeny, you're reconstructing events which happened like maybe before the Cambrium. Yeah. So now the fossils are kind of very well agree what we find in phylogenomics, and things are coming together, which is really nice. So, but this is like still the big backbone. We need the details figured out within the groups. And maybe annelids are even older than you think, because some people now claim that this so-called Claudinomorphs, they are like Claudinia from the Archaea period. It's a period just before the Cambrium, that they maybe represent, in fact, the fossils of annelids with tubes. Yeah, this is highly discussed. As I'm not a paleontologist, I cannot really judge this. And it was like really controversially discussed, this paper. But I see, for example, Rachel Murch on this paper, and she basically perfectly recognizes an annelid tube because she's like the main authority of this field, at least of the present tubes. So this could be interesting to see in the future how old are annelids really, and they might be from the Archaea, which also makes sense because the mollusks, we assume it either way, that they're from Archaea, the first splits. Okay, now I have to check my time. I'm getting later and later. So the question is, what can we use? Because this transcriptomic analyses are often quite expensive. And we were talking about this before briefly. That's something also of how many budget do you have? Can you do this analysis, even though they get more and more inexpensive? What about mitochondrial genomes? Why not use them more? So the 2000s, it was like super difficult to sequence a mitochondrial genome. We were basically making like piece for piece in Sanger and then sticking everything together. And then I spent like half a year, not on this genome, but another, to get my results. And then you had one genome, and I didn't want to do another one. did it with Obinia in the lab, and then later on with another one, but then I was really done. It was quite laborious. But nowadays, this goes much faster. Now I skip this a bit. I don't talk about mesostomids. What we can use is so-called genome skimming. Yeah, genome skimming is a way that you do, instead of transcriptome sequencing, genome sequencing. Yeah, whole genome. Of course, this sounds okay. This is must be more expensive, but you can do this in a very low coverage. That you just say, okay, I want to have like I don't know, 20 million Illumina sequences or 100 million Illumina sequences, which is like really a number of, I don't know, 100 euro, 200 euro what you pay for nowadays, for my taxon of interest. And this will be never enough to reconstruct your genome. But you will have like a lot of other like markers which you can harvest from this data, because some of these organelles or some of these markers are like in a very high copy number in your cell. So for example, every cell has like, I don't know, 100 to 1000 mitochondrial organelles. So the mitochondrial genome is much more higher presented into such data. And the same is true for the 18S or 28S. This is this ribosomal genes, because they are like in tandem, really repeated clusters in the genome. And you have, I don't know, 1000 copies or more, depending on the species, and depending, I guess, also on the cell, of these markers. So even though you cannot get anywhere to reconstruct the genome of your taxon, you can harvest this data for getting the mitochondrial genome. This has been shown again and again. And we use this genome skimming to analyze the relationships of inside one family. It looks like inside one genus here, because in this family, the glycerids, like nearly all the taxa are in the genus Glycera, they all look the same. Yeah. And we use this mitochondrial genomes to analyze this super well. So we sequence low coverage genomic data, and then we harvested the mitogenomes, and now there are many tools online that you directly can analyze your assemblies to get all this data. And then you get the complete mitogenomes, and you have another source of data. You not only have the mitogenomes, but you also have the gene order data, which is maybe another source of phylogenetic markers. This is something what you can do nowadays, very good and quite price effective, or not very costly, to get a lot of data. And these genomes are like 16,000 base pairs to analyze. You get some ribosomal genes on too. So basically you end up with 20,000 base pairs you can perfectly analyze for phylogenetic studies. This might not be enough for deep metazone phylogeny, but it's perfectly enough to work within the families. If you want, for example, to see orbinate relationships, narrative relationships, and so on, because in most of the taxa of the annelids, we don't have very good phylogenetic hypotheses. There are only a few examples which are really well covered now. And of course, when I give a talk in my first talk in Brazil, I also want to show research from Brazil. This has been, for example, done in the group of Cynthia Santos. They did it with like narratives. They worked on the narrative phylogeny, and they used this genome skimming to get the mitochondrial genomes and put them in a tree, analyze them, and basically they could do mitogenomic analysis of annelids. And this is working quite well. And this is also what you perfectly can do, for example, with like a master thesis or so, because analyzing one or several mitochondrial genomes is something what you can easily like do in your graduate studies. While a complete genome is like often a teamwork of like many researchers to really get something meaningful out there. So they're quite well to handle. Okay, but now what is with genomes? What is going on with genomes? Where are the analyte genomes? What is the future? Why now that? So we pay for a human genome $1,000. What do we pay for an analyte genome in good quality? It will be a bit more, I will tell you. But where are they? Where are the analyte genomes? And this is from a review we published 4 years ago. And this is our backbone. And these are like some taxa have genome sequenced. So there were like some old studies like on Capitella telata and Helopdella robusta, which were the first genomes available for annelids. And the data was available 15 years ago. It was formally published, I don't know, 7 years ago. It was forever in analysis. And that's basically it for a long time. Then we have a few other genomes, and now people are doing something. I know the German Martin Duran is working on Ovenia. The Platineres genome is as long announced to come soon, as long as I can think, basically. So I heard this 15 years ago, I heard this 5 years ago, I heard it a year ago. It's still not published, but it will come at one point, the Platinum-based genome. So it's the only model organism without a published genome. It's kind of incredible. And people should definitely look into these guys here, Magellanids and Chaetopterids. So now we can, I don't know, we can get terabytes of sequence data for quite few money. What happened? Where are the genomes? Why are they not coming? And here's a good example. This is the genome of Eisenia foetida, like published 5 years ago, I think, or 6 years. And what they did, they make a lot of Illumina reads, they assemble them into a genome, and then they have a coverage of 40x. It means every position in the genome was covered 40 times, which is like very well. But what they had was a so-called N50 of 1,850 base pairs. So I can tell you, this is like, so it's not really a genome, it's like a jigsaw puzzle. But what happens there? Why do we don't get there? Even though we have such a good coverage, that we have so much data, why can't we reconstruct the genome? And here's some introduction into assembly. So what we want to do is basically want to get context. So what we have, this is what we get from the sequencer. These are reads, and these are short. In Illumina, these reads are still 150 to 250 base pairs generally. Yeah. And then we want to put these little pieces one after another to reconstruct context. Context is nothing else than an analysis of how to put together these reads, and hopefully this resembles part of the genome. And sometimes we can use additional information to have this context into bigger things called scaffolds. So how they're ordered. Yeah. And then we also are interested in the coverage. Yeah. So coverage just means how often is on average every single position of a context read over by a sequence read you get. Yeah. And if this happens 4 times, you have a 4x coverage. If this is not happening, then you wouldn't even put them together in a context, then you have 0 and whatever, yeah? So here in this analysis, every position was 39 times covered. But to assemble these things, you have very short reads, you use so-called Kmere graphs, yeah? So you put them, which is like against your intuition, that you put them in either smaller pieces, the Kmeres, yeah? Kmeres are n-1 overlapping pieces of the same sequence. So these are the different Kmeres of the size 7 of one sequence, what you can see here. And these Kmeres are put together into a graph where they also overlap besides with an n-1. And then you have like a complex Kmere graph. And when you later go over this graph and follow the sequence, you come out with your context, yeah? But finding this context can be problematic because if you have repeats in your genome, basically, if the repeat is longer than this Kmere, we cannot basically like really analyze this repeat regions, yeah? And this happens all the time. Our genomes are full of repeats, yeah? Like the human genome is like I don't know, 50–60% repeat sequences, some transposons, whatever. So it's basically a nightmare for assembly. So we are not getting anywhere with this kind of methods with short reads to reconstruct genomes because we need longer pieces. This is a problem. And we don't get them with Illumina. So what the N50 means, the N50 of an assembly is the weighted median, and half the assembled bases of all contexts are represented by context of this length or longer. That means in this study, half of the contexts explaining the whole data are only 1,850 base pairs and longer. So that's super short snippets. So this is not a genome. But this is always what happens if you just get Illumina short read data and you want to make a genome assembly. And it doesn't matter if you have here 39x or 500x. You're not getting there. Okay, but how we get there? With the third generation of sequencers, and luckily we have like new techniques available. You maybe heard about PacBio and Oxford Nanopore, which are the most prominent ones. And they do single molecule real-time sequences. And they're like single molecules are like either filmed in the PacBio or goes through like a nanopore in the nanopore, and you get much longer sequences, yeah? So instead of that your sequences are 150 base pairs or 200, or with a 454, there were maybe even 1,000 base pairs. We talk about 10,000, 20,000, 30,000 base pairs, and with a nanopore, but these are exceptions. It's like super difficult. There were like sequences in the size of 100,000 and even 1 million base pairs. But this is super difficult to get, yeah. But we are talking about here 10, 20, 30, 40,000 base pair long sequences, and they are perfectly fine and able then to use to reconstruct complete genomes. We did this for a lizard, and we saw like a huge improvement if we only had Illumina, or if we corrected this Illumina with bio and getting longer pieces. And now where you're getting to is, this is like something published this year. It's the same genome, but from a different group, Archaea fetida. And they have a chromosome scale assembly. So basically the number of contacts or scaffolds they have mirrors the numbers of chromosomes this animals have, which is exactly what you would expect, what you want to get if you have the perfect genome. And that's what you are getting now. So we are getting now, but it's just now just starting to chromosome scale genome assemblies. And this has been done for the slittlate annelid Archaea fetida. And I have seen 2 other very, very high quality genomes published this year or last year. This was another, it was a leech, another clitellate, and this is now what I just learned today called Dimorphilus, formerly known as Dinophilus gyrociliatus. It's a myofauna annelid, which is one of the smallest animal genomes. It's only 70 million base pairs long, and these are high quality genomes. But these 3 examples are basically now the exception, but now we are waiting that more genomes are coming. And then, of course, this data can be perfectly used for phylogenetic reconstruction, and also look for other markers. So now if you Google Scholar, look for chromosome scale assembly, you find this is getting now the new norm. Anopheles has been sequenced, even the axolotl, huge genomes, 32 gigabases along on chromosome scale, amazing. But this is what we know. But this also means that all these Illumina genomes, which has been published like in the last 10 years, they basically can be done again, because it makes no sense to analyze them together with this kind of high quality. So that's where we are waiting for. There are many, many initiatives who are working on this chromosome scale genomes. These are old initiatives. Everybody is saying, we sequence all of this, we can sequence all of this. And now, of course, the initiatives are like, we are sequencing all the eukaryotes, or the Darwin tree of life, everything what we find in the UK, which of course is still quite reasonable because you're not finding many species in the UK, as everybody knows, or what we find in Europe. This is new initiatives, the ERGA, the European Reference Genome Atlas, and there's like a proposal that we should try to sequence everything what we find in Europe on a chromosome level to have this data available for biodiversity studies, but also for whatever studies. This is now proposed. In reality, of course, it will be impossible to sample all these organisms, but if you go with such initiative, you start with the most important ones, with the most charismatic ones, with the most used one, and then you go step to step, and let's see where are we getting there. Okay, let's see what the future brings. And thank you for listening, and thank you for the invitation. And I'm happy to take questions when this will be open for questions. Thank you very much. Your mic is muted.
Wow, John.
Thank you very much. So thank you very much, Christoph, for the wonderful talk. And thank you all that have been watching with us on YouTube. I hope you enjoyed as much as we did. Muito obrigada a todos que estavam assistindo com a gente. Espero que tenham gostado. Parece sempre... So now we are back.